I think 14 years ago in 2009, so when I still did postdoc research in Cambridge, under the guidance of Professor Azim Srani and Dr. Kai-Chin Law, we developed the first single-cell RNA sequencing technology in the world. I think the method is quite powerful. So now really many labs are using the single-cell RNA sequencing technology to do many research for different biological or medical studies. And then in 2010, I went back to Beijing to set up my own lab. 3 years later, in 2013, my lab developed the first single-cell DNA methylome sequencing technology in the world. So with this method, you can really identify the DNA methylation patterns in just one individual cell. The method is powerful, but it is expensive because usually if we want to identify really exciting biological features of the methylome, you need the coverage usually around 10%, make it quite expensive. So that's why after 10 years, now still just only several hundred labs using the DNA methylome sequencing technology to address their biological or medical questions. And then in 2020, so my lab developed a high-precision single-cell methylome sequencing method to using the ONT platform. So directly sequence the full-long cDNAs, no matter it's 1,000 base pair long or 3,000 base pair long. And just the sequencing about 20 to 50,000 reads, we can already detect over 3,000 genes within an individual cell and over 4,500 different RNA isoforms. So it's really quite helpful. You don't need help of the 10x NGS platform, just purely based on the ONT platform. You can determine every cell type, the transcription pattern, the RNA isoforms, or even allele-specific gene expression patterns. And then 1 year later, in 2021, my lab developed the first single-cell genome sequencing technology based on the single molecule sequencing platform. So the sequencing length is about 6 kb, and then we realized that after you can sequencing the genome of single cell with kb long read length, it's possible to do the de novo genome assembly. So last year, so my lab saw that just sequencing as minimal as 30 single cells genome, and then we can do the de novo assembling to get the human genomes contig with N50 more than 1.3 meg-base long. So with this meg-base long contigs, it's very easy to identify all of the exciting features of our genome. And these work are already some years ago, so today I will not talking about them. Today I will mainly talking about a single-cell NanoATAC sequencing technology my lab developed recently. And as you know, 8 years ago in 2015, Will Greenleaf's lab developed a single-cell chromatin accessibility sequencing technology they call the scNanoATAC-seq method. It's really a beautiful method because it's both powerful and cheap, because we know that the open chromatin feature or the chromatin accessibility region in our genome is relatively small. It accounts only less than 1% of our genome. That means you only need to sequence about 20 to 50,000 reads for each individual cell. You can already clearly discriminate different cell types. And then when you merge about 50 to 100 cells together, you can beautifully identify all of the open chromatin peaks within the cell type. So now, just after 8 years, the scNanoATAC-seq method really now I think tens of thousands of labs are using it to identify the epigenetic layers, the open chromatin feature, and how it regulates gene transcription. And we know the open chromatin region in our genome usually are quite short, just between 100 to 300 base pair long. And then, so why consider to using the ONT platform, using the thousands base pairs long reads to do these so short fragments? So most people think it's not feasible or it's not useful, but we have some different idea. We saw that, in fact, it is possible to using the ONT sequencing platform to do the single-cell ATAC-seq analysis. So the idea is like this. So first, although the open chromatin region is less than 300 base pair long, so Will Greenleaf so that you use the TN5 transposase to cutting within this 300 base pair long fragment twice, and then you amplify the intermediate DNA fragment, and then do the sequencing on the NGS platform. But we realized that, in fact, when you adjust the TN5 activity properly, and you cut within these 300 base pair open chromatin regions once or twice consistently, and also several kb away, it will also relatively randomly cutting between two neighboring nucleosomes, even it is closed chromatin region. Because even at closed chromatin region, between the neighboring nucleosome, there's still around 30 BP relatively free or naked DNA region. And then we can amplify these several kb long DNA fragments. And for each end of the fragment, we do the pig calling, and then we can easily identify all the open chromatin features. But now our relance extend from the NGS forms 300 base pairs to about 8,000 base pairs. And then we can identify a lot of heterozygous SNPs, and they give us a lot of exciting new powers. And we first saw that, in fact, our method can comparably get comparable achievement compared to the single-cell ATAC-seq method. For example, for the gene promoter region, you can clearly identify the open chromatin feature around the TSS region. And for different cell types, we can identify the footprint of the transcription factors, because we know transcription factors are really master regulator to determine the cell's fate. And you can see around the motif of the CTCF transcription factor, there's a valley on the peak. That's a footprint, means the CTCF protein just binding to the motif and make it a little less accessible. And when we using the bulk ATAC-seq result from the GM cell line, and you can see our single-cell NanoATAC-seq method can achieve comparable detection power compared to the single-cell ATAC-seq method. On the right side, you can also see we can nicely call the open chromatin peaks comparable to the single-cell ATAC-seq. And when we apply the method to different cell type, we can really determine the cell type specific transcription factor binding features and the footprints. And when we mix a lot of cells together to do the single-cell sequencing, we saw our method have quite low DNA doublets. So that means there's minimal cross-contaminations between individual cells to permit us to determine the cell type accurately. And when we just sequencing every single cell, only about 20 to 50,000 reads, we can already clearly discriminate all the 5 different types of cells ambiguously. And we know for many studies, for example, for cancer study, when you do the single-cell analysis, you want to clearly discriminate the aneuploidy cancer cells and the euploidy, for example, epithelial cells. So we saw that our method can achieve that. On this figure, you can see in the K562 cancer cell line, we can clearly identify these chromosome arm levels copy number variation, no matter it's copy gain or copy loss. And compared to the control of the EHAP1 cell, you can see it's a euploid cell line, and basically there is no copy number variations. So it means there's minimal background when we're calling the copy number variation using our method in every individual cell. And important thing for this single-cell ATAC-seq method is to identify the allele-specific open chromatin feature, because we know for majority of our human cells is diploid. So for every gene, there's 2 copies or 2 alleles. So sometimes, or quite sometimes, in fact, the 2 alleles open chromatin feature can be asymmetric. It can be the maternal allele is fully open, the paternal allele is fully closed, or vice versa. Or maybe one of the allele is strongly open, the other allele is weakly open. So if you're using the William Greenleaf developed single-cell ATAC-seq method, if we want to discriminate the 2 alleles open chromatin difference, so you need to, within the 300 base pair open chromatin region, at least 1 heterozygous SNP. But for our method, we don't need a heterozygous SNP within the 300 base pair. We only need it within the neighboring 8,000 base pair, as if you have 1 heterozygous SNP. That's enough to permit us to determine the allele-specific open chromatin feature. So that means, in general, we can amplify the efficiency to detect allele-specific open chromatin feature for more than 20-fold compared to the William Greenleaf's method. So from this figure, you can see basically the paternal allele is strongly open, whereas the maternal allele in red is weakly open. And we apply this method to a cell line, the GM cell line, and identify a lot of these allele-specific open chromatin features. And you can clearly see majority of them enriched on the X chromosome. This perfectly matches to the known biological feature of this cell line. It's a female cell line. Every cell contains 2 X chromosomes, and one of the X chromosomes will be randomly activated, inactivated. But for this cell line, the inactivation is not fully random, but strongly biased to silence preferably the paternal allele. And you can see for majority of these allele-specific open chromatin feature we identified on the X chromosome is mainly open on the paternal allele, on the maternal allele, but closed on the paternal allele. So perfectly matched to previously known knowledge about this cell line. And then we can show some examples for the imprinting gene, because we know the imprinting gene is very epigenetic regulated, because it can be identical sequence, but the paternal allele is fully open, whereas the maternal allele is fully silenced, or the vice versa. So for this imprinting gene, the SNU13, it is known that the paternal allele is open, whereas the maternal allele is fully closed. But in this GM cell line, so basically within this opening peak, the 300 base pair, there is no heterozygous SNP. So we cannot determine if it have this allele biased open chromatin feature, or we don't know if it will be imprinted using the single-cell ATAC-seq method. But when using our single-cell NanoATAC method, we sequencing the neighboring 8,000 base pair region, so we can easily identify the heterozygous SNP. So from the figure, you can see the paternal allele is fully open, the maternal allele is fully closed. So exactly match the known situation for this imprinting locus. For another imprinting gene, the TRIM61, so it's the opposite pattern. So you can see the paternal allele is fully closed, the maternal allele is fully open. But again, within this 300 base pair open chromatin region, there is no heterozygous SNP. So the Will's single-cell ATAC-seq method cannot determine if it is imprintingly open or not. But our method, within the neighboring 8,000 pairs, we can easily identify the heterozygous SNP to show that the paternal allele is fully closed, the maternal allele is fully open. And our method, we can not only detect the chromatin accessibility, but some of the reads just randomly fragmented from the genome. So that means we can also detect the single-cell genome, mainly the structure variation, because we know the NGS platform-based single-cell sequencing method, it's very difficult to detect these structure variations with a length between 50 base pairs to 10,000 base pairs. But our method, you can see, we can not only detect the chromatin accessibility, the epigenetic information, but also the genome information. For example, for the structure variation events, including the inversions, deletions, we can identify them very accurately. And one type of the structure variation is more difficult to detect, the translocation. So the NGS platform-based single-cell sequencing is very difficult to detect them. But for our method, it's very easy to detect. So for this K562 cell line, you can see there is a translocation and make a gene infusion event between the BCR-ABL1 gene. And you can see we can clearly identify it. And for the insertion event, for example, 300 base pair insertion, and even 89 KB deletion event, we can clearly identify them. You can see 2 of the genes are just deleted due to this event. So our method can detect both the epigenetic chromatin accessibility and the genomic information, including structure variations. And then for the single-cell ATAC-seq, another powerful use is to detect the co-opening of the different genomic regulatory elements, for example, the enhancer and the promoter. So usually, if an enhancer need to regulate a promoter, So it need to be open, both the enhancer and the promoter, and then through the loop extrusion, they become near, and then the enhancer will activate the promoter and make the transcription started. But when we're using the Will's single-cell ATAC-seq method to detect this co-opening, there are about 50% of this co-opening will be artifact, because we know these co-opening need to be insist on the same allele. So that means if enhancer on the paternal allele open, it will not affect a promoter on the maternal allele. It must be on the same allele. It will just regulate it. But using the Will's single-cell ATAC-seq method, 50% will be this way, not this way. But our method, in fact, we directly sequence the connecting DNA fragment within the promoter and the neighboring enhancer. So as if we detected, it must be on the same allele. So totally no any artifacts. So in this way, we saw that we can detect many these co-opening features between the enhancer and the promoter, or enhancer and the enhancer. And here are some examples. You can see for this SOX4 gene, the enhancer and promoter are beautifully co-opening nearly in every individual cells. Another example is about this histone genes cluster. So many copies of different histone genes here, and you can see either is enhancer promoter or even promoter are co-opening within every individual cell. So meaning that it must be enhancer regulating the promoter, or the promoter are interacting and regulating each other's activity. So after we saw that the method worked beautifully in the cell lines, so we want to address some real biological question we interested. My lab quite interested in this pre-implantation embryonic development. So we're using the method to analyze the mouse pre-implantation embryonic development from the single-cell zygote stage until the late blastocyst stage. And for every stage of the embryo, in every embryo, it only contains very few cells. Even at the late blastocyst stage, only about 60 cells in one embryo. So if you don't using the single-cell approach, it's very difficult to analyze. And also, we know that in the late blastocyst stage, there are already 3 different types of cells mixed together. So if you don't use a single-cell approach, you cannot discriminate them properly. And first, we saw that after we do the single-cell NanoATAC-seq for these mouse pre-implantation embryos, you can see the beautiful developmental path of the embryo, starting from the single-cell zygote stage, go through the 2-cell stage, 4-cell stage, 8-cell stage, go to the modular stage, and then to the blastocyst stage. First, it separated into the trophectoderm and the inner cell mass. And later, the inner cell mass will further separate into the epiblast and the perimeter endoderm. So it's really a beautiful developmental path for the first time we can capture. And after we can capture the path, now we know that we can determine for every different stage of the cells to see the heterogeneity of the development. You can see, for example, for the modular stage, the cells are relatively synchronizedly developed. So most of the cells are at a similar open epigenetic state. But if you're looking at the A-cell stage embryo, there's a lot of heterogeneity. Some cells development is strongly delayed compared to others. So purposely, if you merge the cells using bulk sequencing, you cannot see this heterogeneity. So you cannot not understand why even in the same development stage, the embryos or the cells have some different developmental state. So now we can really decipher these things clearly. And I think now with this single-cell NanoATAC sequencing technology, now we can really identify the dark matter in our epigenome. Because previously we know that based on the NGS platform, when we do the single-cell epigenome sequencing, for these structural variations or repetitive elements, we can just not detect them. Because when we're mapping the sequencing rate, it will not work for most of these structural variations or the repetitive elements. If you cannot mapping, you don't know if these regions have any open chromatin feature or closed chromatin feature. But now with our method, we can clearly identify these structural variations and mapping easily to these repetitive elements. And then we will know how many of them are in the open chromatin state, how many of them are in the closed chromatin state. and how it contributes to the cell fate determination. And finally, I'm to thank all my students in my lab, especially Yuqiong, Zhenhuan, and Kexuan, to developing this method. And thank you for your attention.